Hello everyone, our today's topic is fluid hemodynamics. Now we know that the life it has a origin in the sea. Many scientists they believe that the life it originated in the oceans. As the early earth, remember they have right conditions for the formation of simple organic molecules. Similarly, our body are similar in composition as compared to the sea water. Comparing the cell environment as similar as ocean water is a simple way to understand that. The cell environment, remember, the cell environment it includes fluids, cells, fibers. Apart from that, it includes the ground substance. The ground substance that will provide the structural support and helps to mediate the biochemical interaction. Now the term internal environment, it was first described by Claude Burnett. So according to him, he introduced the concept of internal environment. He described it as a stable condition that is necessary for survival of the cells within an organism. So it refers to the interstitial fluid and plasma maintaining the cell and tissue function. So this internal environment it consists of interstitial fluid, that is the fluid that surrounds the cell, and plasma, that is the liquid component of the blood. So this fluid will provide a consistent environment that will support the normal function and the structure of the cells and tissue. Now, what is a homeostasis? The process of maintaining internal environment stability is homeostasis. So, remember homeostasis, it is a mechanism by which the body what it does is it tries to maintain a stable internal environment. So the body tries to maintain stable internal environment despite there are changes in the external conditions. Now we know that the water is the main component in the body. We also know that water is the vital component of our body. This water it acts as a solvent for biochemical reactions, it acts as a medium for transporting the nutrients, and even it helps to maintain the body temperature. So remember the water it make up around 50 to 70% of normal adult body weight, that is about 60% on average. Whereas females, remember, they have a lower percentage of body water as compared to male. Now the water distribution in the body, the water can be distributed as intracellular fluid compartment or extracellular fluid compartment. The intracellular fluid compartment, remember, it forms around 33% of body weight. So intracellular fluid, it is the fluid that is present within the cells, and it is present mainly in the muscles. Whereas the extracellular fluid, it composes of 27% of the body weight, and it is the fluid that is present outside the cells and it can be divided into various subcomponents. Like the first is the interstitial fluid. The interstitial fluid, remember, it forms the major part of extracellular fluid. So interstitial fluid is the fluid between the cells in the tissue, and it is formed of 12% of the body weight. The next is intravascular fluid, also known as blood plasma, and we know that the blood plasma, it's the liquid component of the blood, and it forms 5% of the body weight. Remember, plasma is about 3 liters, and out of which 5 liter is the blood. The third component is the mesenchymal tissues, that includes the dense connective tissue, cartilage, and the bone. So since these tissue will contain water, and it forms around 9% of the body weight. The last are the transcellular fluid. It is in a very small amount, that is only 1% of the body weight, and it includes the secretion from glands and the CSF fluid, that is cerebrospinal fluid. Coming to the electrolytes that are present in the body fluid, there could be intracellular or extracellular. In the intracellular fluid, the main cation that is present is potassium. Potassium is the main intracellular cation, and it accounts for the 98% of total body potassium. We know that potassium, it helps to maintain the resting membrane potential of the cells, and that is very important for the nerve transmission, muscle contraction, and overall cellular function. The magnesium is another important intracellular cation. It plays a role in energy metabolism, that is ATP production. Apart from that, protein synthesis and muscle and nerve function. The extracellular fluid, the predominant cations, they are sodium. Sodium is the major extracellular cation, and it accounts for 90% of all cations that are present in extracellular fluid. It helps to regulate blood pressure, facilitate the nerve impulse transmission, and it helps in muscle contraction. Other anions, principal anions, are chloride. Chloride helps to maintain electrical neutrality and osmotic pressure. Apart from that, we have bicarbonates. Bicarbonates, they are the major buffering acids in the blood and help to maintain the acid-base balance in the extracellular fluid. Now, what are the major functions of electrolyte? First is they help to maintain the acid-base equilibrium. The bicarbonates and phosphates, they act as a buffer. That means they stabilize the pH, and by accepting or donating hydrogen ions, they help to regulate the acidity or alkalinity of the body fluids. Second is it helps in regulation of osmolality and body and the fluid volume. Now let us discuss the component of normal fluid pressure. The first is osmotic pressure. It can be divided into two. One is crystalloid osmotic pressure and other is colloid osmotic pressure. The crystalloid osmotic pressure is exerted by the electrolytes, that means the ions that are dissolved in extracellular fluid. So this crystalloid osmotic pressure, it helps to maintain the water balance between the extracellular and the intracellular compartment. Whereas the colloid osmotic pressure, it is exerted by proteins like albumin that is present in the plasma. We know that due to the higher protein concentration in the plasma as compared to the interstitial fluid, the osmotic pressure of the plasma, we know it's higher than the interstitial. Plasma pressure is 25 mm of mercury and interstitial pressure is 8 mm of mercury. So the effective oncotic pressure is the difference between these two pressure, and that is important for drawing the fluid from the interstitial space back into the capillaries. Other is hydrostatic pressure, that is the capillary blood pressure. That has, we know capillary, they have two ends. One is the arteriolar end and other is the venous end. So hydrostatic pressure, it is the pressure that is exerted by the blood. So suppose these are the capillaries and blood is passing, we know, through them, and it is the pressure that is exerted by the blood against the walls of the capillaries, and that is hydrostatic pressure. And remember, this pressure would vary with the length of these capillaries. So the arteriolar end, the hydrostatic pressure is higher. That means that facilitate the outward movement of the fluid and nutrients from the capillaries into the interstitial space. Whereas in the venular end, the hydrostatic pressure it's lower, an average of 12 mm of mercury. That will facilitate the reabsorption of fluid back into the capillaries. Another pressure is interstitial fluid hydrostatic pressure. It refers to the pressure that is exerted by the interstitial fluid against the capillary walls. So it is generally lower, that is 4 mm of mercury, as compared to hydrostatic pressure that is present within the capillaries. So the effective oncotic pressure, it is the net difference between the higher oncotic pressure of the plasma and the lower oncotic pressure of the interstitial fluid. So this pressure would act as a force. So this force would draw fluid from the interstitial space into the capillaries. And net hydrostatic pressure, it is the difference between higher hydrostatic pressure in the capillaries and lower tension tissue interstitial fluid pressure that helps to draw the fluid from the capillaries into the interstitial space. So the balance between hydrostatic and oncotic pressure, they are exchanged across the capillary walls, and this pressure it is important for delivering the nutrients removing the hydration, maintaining the proper hydration of the tissue, and overall tissue function. Now remember the imbalance in these pressure that would lead to edema formation. That means there would be excess of fluid in the tissue. So disorders like hypoalbuminuria can decrease the oncotic pressure and lead to edema formation. Now this diagram illustrates the mechanism that is involved in formation of edema with the accumulation of excess fluid in the body. The first is the normal fluid exchange. So at arteriolar end, you will see here the pressure is 32 mm of mercury and the oncotic pressure here is 25 mm of mercury. So the hydrostatic pressure, this hydrostatic pressure, it will pushes the fluid out of the capillaries, while the oncotic pressure pull the fluids into the capillaries due to the presence of plasma protein. At the venular end, you will see the hydrostatic pressure, it drops to 12 mm of mercury, whereas the oncotic pressure remains same, that is 25 mm of mercury. So the lower hydrostatic pressure allows fluid to re-enter the capillaries from the interstitial space. So under normal condition, the balance between these pressure ensures that no excess fluid accumulates. And there is the lymphatic outflow. It helps to remove any excess fluid. The option B is when there is a decrease in plasma Option B is when there is decrease in plasma oncotic pressure. So when the plasma protein are low, that means there is hypoproteinemia. That means here the oncotic pressure would decrease. That lead to edema. So when the plasma proteins they are low, the oncotic pressure decreases. Lower oncotic pressure means that less fluid is pulled back into the capillaries from the interstitial space that lead in fluid accumulation. Option C is Option C is capillary pressure. So increased hydrostatic pressure in the capillaries, it can cause edema. If the hydrostatic pressure in the capillaries is increased, it forces more fluid out of the capillaries into the interstitial space. So the higher hydrostatic pressure, it causes imbalance, lead to fluid accumulation in the tissue. Option D is lymphedema. That means when there is lymphatic obstruction. So when the lymphatic vessel they get obstructed, the fluid that is normally removed from interstitial space, they cannot be drained. So that lead to accumulation of fluid and the excess fluid that will not be drained that lead to lymphedema, that is the fluid accumulation. Option E is tissue factor. So this describe how tissue factor can influence the development of edema. So increase in interstitial oncotic pressure due to presence of more protein in the interstitial space that will pull more fluid out of capillaries. Reduced tissue tension can also contribute to fluid accumulation in the interstitial space. So both factor would lead to edema formation. Option F is capillary permeability. How increased capillary permeability can cause edema? When the capillary walls they become more permeable, more fluid leak out into the interstitial space that lead to imbalance and lead fluid accumulation or edema formation. Now the option A, remember, it's a normal condition when there is balance of pressure and there is no edema formation. Option B is when there is decreased plasma oncotic pressure. So decreased plasma oncotic pressure would lead to less fluid resorption and formation of edema. Option C is increased capillary hydrostatic pressure that force more fluid out that lead to edema in the tissues. Option D is lymphedema. Obstruction of the lymphatic drainage that would cause fluid buildup. Option E is changing in the tissue factor that would affect the fluid movement. And option F is increased capillary permeability that will allow more fluid to leak into the tissues. So leading to ultimately edema. Edema means abnormal accumulation of excess fluid in the serous cavity or in the interstitial space. Now these fluid can be free fluid in the body cavities that could be either ascites, when fluid accumulates in the peritoneal cavity, that is the abdominal cavity that surrounds the organ like liver, intestine, ovaries, or hydrothorax. Fluid build in the pleural cavity surrounding the lungs, like the congestive heart failure, pneumonia, etc. Or pericardial effusion, accumulation of fluid in the pericardial cavity around the heart. Now these fluid can also accumulate in the interstitial space, that is the between the cells in the interstitial space, that can lead to pitting or non-pitting edema. So non-pitting edema is seen in hypothyroidism or during the parasitic infection, like in case of elephantiasis. Now this fluid can be localized over one area, or it can be generalized. Generalized edema is also known as anasarca. The fluid can be transudate or exudate. Transudate is typically low in protein and results from increased hydrostatic pressure. Or decreased oncotic pressure, and it is seen in congestive heart failure or liver cirrhosis. Whereas exudate, remember, it is a inflammatory type. So there would be higher protein content, increased vascular permeability, and there would be inflammation. And it is seen in like rheumatoid arthritis. Now this one is the difference between exudate and transudate and exudate. The transudate, it results from filtration of blood plasma through the normal intact capillaries where there is no significant change in endothelial permeability, whereas the exudate, it arises from the increased vascular permeability in the inflamed tissues that allows protein and cells to leak into the interstitial space. So typically, transudate is non-inflammatory, whereas exudate, it is inflammatory. There would be presence of inflammatory cells and mediators. The protein content is low in transudate. It lacks significant fibrinogens and coagulation factors, whereas exudate has a higher protein content with elevated fibrinogen, and it has ability to coagulate. The glucose content is same as that in plasma, whereas exudate shows a decrease in glucose level as compared to plasma due to consumption by the inflammatory cells. Transudate has low specific gravity due to its lower protein content, whereas exudate has higher specific gravity due to higher protein content and cellular component. The pH, transudate has a pH above 7.3, whereas exudate, it has pH below 7.3 as due to presence of inflammatory byproduct. The lactate dehydrogenase, the LDH level, they are low in transudate and they are increased in exudate, that indicate cellular damage and breakdown. Transudate, it is seen in condition like congestive heart failure where fluid will accumulate due to hemodynamic changes, whereas in exudate it is seen in purulent infection where pus formation results in thick cellular rich fluid.